How are you all? Now tell me, how many of you like to read storybooks? Good. So you all know that every story is framed with paragraphs which are made up of sentences and each sentence is made up of words. Children, can you tell me what does a word made up of? Exactly, letters. So, letters are the building blocks of sentences. Our topic today is also related to such building blocks. Take a look at the picture on the screen. Can you tell me what material is required for the construction of a building? Yes, we need bricks, cement, iron frames, and a plan. Using bricks and cement, rooms are constructed as per the layout of the plan. Then, what are the building blocks of a house? Yes, bricks are the building blocks of any house. Similarly, all the living organisms, be it small ants, grass, dogs, fish, or big trees, including our cells, are also made up of small units. These are called cells. Cells are the basic structural units of all living bodies. Alright, now if I ask you to distinguish between living and non-living things, you will answer immediately that living organisms can breathe, reproduce, move, respond to stimuli, etc. But did you ever think how living organisms could manage these functions which cannot be performed by non-living things? Before learning more about cells, let's learn about their discovery first. The invention of magnifying lenses led to the discovery of the microscopic world. The history of cell journey dates back to 17th century. Robert Hooke, an English philosopher, architect, and a polymath, in 1665 discovered and named the cells while examining a thin slice of cork under a self-designed microscope. Children, do you know what a cork is and how does it look? Cork is a porous substance obtained from the bark of a tree. Robert Hooke observed several honeycomb-like chambers which are empty and surrounded by walls resembling monasteries. Robert Hooke named them cells, which means a little room in Latin. Later, a Dutch lens maker and also a cloth merchant, Anton van Leeuwenhoek, in 1674 discovered the free living cells in pond water for the first time. He named them animalcules. Can you think of a reason why Leeuwenhoek called them animalcules? It is because they were freely moving in the medium like small animals. Leeuwenhoek, during his lifetime, prepared 200 lenses of different focal lengths with which he used to observe the fine texture of cloth. By the way, he also discovered sperm cells and also identified the sperm cells of humans, dogs, rabbits, frogs, fish, and insects. Children, I hope you are noting down the points. I will check it up in the class. Going back to Robert Hooke, he had only seen the thickened walls of the cells and no substance within them. But in 1831, a Scottish botanist, Robert Brown, stained a dark body in the center of the cell and named the nucleus. Subsequently, in 1839, J. E. Perkin observed a living fluid substance and called it protoplasm. During the same period, two renowned scientists proposed the cell theory. In 1938, Jacob Matthias Sladen, a German botanist, first proposed the idea that all plants consist of cells. A year later, in 1839, Theodor Schwann, a German zoologist, independently asserted that all animals and plants are made up of cells. This joint finding forms the basis of the cell theory. The cell theory was further refined in 1855 when the German biologist Rudolf Virchow presented the idea that omnicellulae acellula, which means all the cells arise from pre-existing cells. Following are the postulates of the cell theory. Number 1. All organisms are made up of cells and their components. Number 2. All the functions performed by the organisms are the resultant biochemical reactions of the cells. Number 3. The cell is the basic structural and functional unit of an organism. And number 4. All cells are formed from pre-existing cells. Hope you all understood the facts. Now, try and answer the following. Why were the chambers observed by Robert Hooke are empty? What was the discovery of Robert Brown? What was the last statement of cell theory and who proposed it? Dear children, as I explained earlier, the invention of microscope during the 17th century was instrumental in the discovery of cell structure and its components. Let's have a look at the development of the microscope. First of all, what is a microscope? Do you have any idea? A microscope is an instrument used to magnify objects. Structures that can't be seen by the naked eye can be observed under a microscope. Microscopes are of three types. Number 1, simple microscope. A single convex lens and can magnify the object up to 10 times. Number 2, compound microscope. This microscope consists of 2 or more lenses and illuminated by light. So, These microscopes are also called a light microscope. The observer views the object or specimen through the two magnifying lenses, the eyepiece and objective lens. Children, did you identify both the lenses in the picture? By using this microscope, we can magnify the object 300 to 1500 times. You might have seen this microscope in our school laboratory. And number 3, electron microscope. As shown in the picture, it is a very large instrument that uses electromagnets for magnification and electrons for illumination. This amazing instrument was developed by Ruska and Noll of Germany in 1940. It uses very high voltage electricity. Children, do you know that we can magnify the objects 1 lakh to 5 lakh times? The structure of most of the cell organelles such as endoplasmic reticulum, mitochondria, chloroplast, nucleus, etc. were known and understood only after the discovery of an electron microscope. Hope you all understood the concept and enjoyed it. So, before winding up the session, let me go back to the question that is posed at the beginning of the video. That is, what makes the difference between living and non-living things in performing functions though they are made up of atoms and molecules? The structural composition of cells, their components and their organization are made able to possess such characteristics. Let's know about it all in the next session.